Hi, welcome to the lab. We're doing nervous tissue and the brain this week. By the time you get this, there should be another assignment down here, which you can access on the brain tissue report. So you're going to have, brain tissue and brain lab page. So you will have a lab report plus an assignment that you will do in mastering. As I said, they will both be linked to here. There's your lab report already linked, and then once mastering is done syncing with Canvas, they still need to talk to each other. That assignment will be posted here. I have taken the objectives from both of the lab guides and compiled them here and made a couple of alterations. So when it comes to the connective tissue and the nerves, we're only knowing the description. We are skipping cerebrum, cerebellum, and spinal cord. And you will see as we go through the lab guide itself that there's a few other things that I've just eliminated. So if you click on here, you will get... Ooh, I've got two files open. Which one do I want? You will get this file here. And as you can see here, for the connective tissue, it says just definitions, and then I've eliminated these three. So we're doing both the histology of nervous tissue and the brain. It's a fair amount of information, but I don't think it'll be like a brutal lab. So I'm just going to walk you through the basics of what you need to know. Starting with, what do we want to start with? I was going to start with the activities. So we'll skip past all of this, and oops, I almost went past them. Activities. So histology, it is multipolar neuron, and then we're not doing the cross-section of the medullated nerve, just the definitions. The picture of the multipolar neuron in the lab guide is pretty good. If you want more pictures, you can go to Practice Anatomy Lab. If you do histology and nervous tissue, right, those multipolar neurons there look a heck of a lot like the multipolar neuron in your lab book. So you can just scroll through. They have some zoomed in way more than we can get with our microscopes. And that one there, just beautiful. So as you can see, right, there's the cell body, and it has multiple dendrites coming off of it, plus an axon. So many processes, multipolar. If you're curious about anything else, there is... they do have the cross-section of the nerve in here. I just don't like it, so I'm not going to make you learn it. And they have cerebellum slides, but not cerebrum. And we're going to have a slide for spinal cord that'll look like this when we do the spinal cord next week. So that's why we're skipping that stuff. So going back to our lab guide then, be able to identify a multipolar neuron when you see one, and then know these three different tissue layer types. So it's very much like a muscle, right? If this whole thing is the nerve, then the connective tissue holding the whole thing together is epineurium. Then for neurons, we are going to call an axon a nerve fiber. So that's the long cylindrical part of the neuron. And just like long cylindrical muscle cells we call muscle fibers, neurons we can call nerve fibers. And nerve fibers or neurons are bundled into fascicles the same way muscles would be. So here's 1, 2, 3, 4 fascicles. Around each fascicle is perineurium. And then if you zoom into one individual fascicle, and now that's an axon, that's an axon. Oh no, I'm sorry. Where the heck is it? There's your endoneurium there. They don't do a good job of showing you endoneurium. They should have a blow up of a fascicle so you can see endoneurium. But endoneurium would be the connective tissue in between the individual, or excuse me, around each individual axon or nerve fiber, but these are all bad pictures. So just know the definitions is what it boils down to. While we are talking about this, there is one thing that I want to make clear, and I forgot to give myself, whoa, what did I just do there? Some white space to do it in, and I'm just going to freehand all of this. That is the difference between a neuron and a nerve. So this doesn't show up in lecture, but it is something you should know. So a neuron is for one-way communication. Right? If the cell body's here, there's your axon, axon terminal, whoop, whoop, whoop. Yeah, pathetic, I know. I'll even give it a nucleus. Right? If you are a neuron, action potentials always only travel in one direction. So a neuron is either carrying information away from the brain, and it is a motor neuron, or it's carrying information to the brain, so it is a sensory neuron. A nerve, because as we saw here, right? Oh, here we are, right? Each one of these little circular things is an individual neuron. So you've got many, 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 many axons in a nerve, which means a nerve is, first and foremost, many neurons. So it is almost always bidirectional. Did I spell that right? Direction. Pretend that's a C. Channel. Which means, in terms of is it sensory or motor, it's mixed. So most nerves contain both sensory and motor neurons. There are a few, like your optic nerve, that are completely sensory. But I can't think of any nerve that is 100% a motor nerve. Primarily because your brain requires feedback from muscles so that it knows what the tension is, so that it can feel what a contraction feels like. So your brain needs to feel what your body is doing so that your brain knows that it's done the right thing. This is all part of learning coordination and how to use your body. All the sensory feedback from your muscles is what allows your brain to know what it feels like when you've done the right thing, so that you can repeat the stroke when you finally hit a good drive playing golf, that sort of thing. So generally speaking, almost every nerve in your body contains both sensory and motor neurons. To complete my analogy, if there is a nerve that's replying motor instruction to a muscle, it is also most likely going to contain the sensory fibers coming back from that nerve. If you have a nerve that goes from the spinal cord to a muscle, why not have it contain both the sensory and the motor neurons? It's just more efficient that way. So do keep that straight. Let's see, so we did neuron, that stuff. Then on a neuron cell model, you want to be able to identify all of these things, and you want to know their function if they have a special function. And mostly I'm thinking dendrites, axons, axon hillock, and the axon terminal. Right? The dendrites being the receptive regions where the neurotransmitter receptors are. Axons carry action potentials. The axon hillock is where the action potential originates. Synaptic knobs or terminals release neurotransmitter. And the myelin speeds up action potentials or increases conduction velocity. So that should all be stuff that's in your lecture notes. Make sure you refer to that when you're studying the neuron model. If you want to know where to find a good neuron model to study, there is a good one in Practice Anatomy Lab, and I also stole a picture of it for you. Don't tell the good people at Pearson that I did that. Anatomical models, nervous system. Ooh, where did it go? I think it's all the way at the end of here, which is why I went and just stole a picture of it for you. Oh, no, now I don't know where it is. Well, this is why I stole a picture of it, because it was difficult to find. Right? Oh, this is embarrassing. I'm videoing myself, and I can't find the model. But it would be in the nervous system. Maybe it's... Let me just look peripheral nervous system real quick, see if it's there. There's a brain. That's not peripheral. Oh, it's there for your cranial nerves. That's why. There it is, part of your peripheral nervous system. But I have included this PowerPoint, which I just dumped a blank slide into so I could draw. Here is my version of what the book has. I added myelin, node of Ranvier, and I put the axon label here. So I'm going to call this the axon hillock. On Practice Anatomy Lab, it's just the axon, and then I just label the axon down here to differentiate the hillock from the rest of it. When you are looking at this model, what you need to understand is that it's cut off here, so you cannot see the axon terminal of this neuron. So what they have done, which the same thing has been done with the model that we have in our lab, only the colors are a little bit different, is these orange things here are supposed to be the axon terminal of neurons somewhere else that are dumping neurotransmitter onto this neuron. So you want to imagine that these are all the ends of axons from other neurons. And this is the postsynaptic neuron for all of these synapses. And I didn't need to draw all that stuff. But I got my pen out, so I want to. So you have the labeled picture with the labels I would use. I put the node of Ranvier in there too. So you'll know the way I'm going to organize this. If I ask you, if I point to the outside, either here or here, I just want you to say Schwann cell. If I ask you what's inside the Schwann cell, I want you to say myelin, and then the gap in between the Schwann cells are the nodes of Ranvier. So there it is labeled. There it is, unlabeled, printed out, practice with it. That is my gift to you. Let's see. So yeah, to know all those functions and that. So then we get on to gross anatomy of the brain, which will not be as bad as it sounds. It is a fair amount of new information, but it's not crazy. Let me find the objectives, because I did cross a few things off for you. We just include so many pictures in these lab guides. So actually I didn't cross much off. If you look at this, what I crossed off was part of the cerebellum, so you still need to identify the cerebellum, just not the white matter in the middle. And then for these two things, diencephalon is a region of the brain that includes the thalamus, hypothalamus, and epithalamus. But I'm not going to have you identify the diencephalon, just the individual parts. Same thing, brainstem includes these three parts, so be able to identify the parts, but don't worry about identifying the brainstem itself. So the lobes are pretty straightforward. They're each just named for the bone to which they are deep. So the frontal lobe behind the frontal bone, parietal, temporal, occipital. We have an image for that. But then there are fissures, sulci, and gyri. If we go up here, which one? So these deep grooves, this longitudinal fissure here, runs very deep down into the brain. You can't see from that cross section, but here you can see it goes pretty deep. So the deep grooves in between parts of the brain are called fissure. There's your longitudinal fissure. There's your transverse fissure, which separates the cerebrum from the cerebellum. Then you have sulci and gyri. So you will notice that the brain just looks sort of lumpy and maybe like a pile of worms that somebody put in a jello mold. Each little thing that would be the worm is a gyrus, and then the little grooves in between the worms are sulci. So if we look down here, this is your lateral sulcus, which separates temporal lobe from parietal and occipital. Then you have this groove here that is the only one that runs from the transverse fissure all the way down to the lateral sulcus. You can spot it up here. It's, oh no, it's this one. I'm sorry. Whoop. They have it a little bit different, but it's this one because it goes all the way down to the lateral sulcus. That separates your frontal lobe from your parietal lobe, and it also separates these two bumps here that we care about. Your precentral gyrus is this lump here, and your postcentral gyrus is this lump here. We are going to identify them in lab because they both contain important parts of your cortex. And by cortex, I mean, whoops, sorry, I'm skipping around. Right, all of this, I won't draw on it, but everything outside of this stuff here, this is all cerebrum. But you've got gray matter and you've got white matter. The gray matter is what's called cortex, and that's where stuff happens. That's where your motor instructions come from, and that's where your most of your sensation takes place. So it's the gray matter or the cortex of your precentral gyrus and postcentral gyrus that include two important and easily identifiable parts of the brain. So let me just go back to the list because I'm losing my space in terms of what I've talked about and what I haven't. So we got lobes, easy. Fissures, there's the two. Sulci, we know what a sulcus is now. We're not worried about the one that I crossed off. Those two gyruses. So then it's just the rest of the brain, which should be straightforward. Where do they have it all labeled? Oh, well, they do it weird. They don't label everything here on the same. So thalamus, hypothalamus, midbrain, pons, and medulla. So if you go down here, this is thalamus, this is hypothalamus, midbrain, pons, and medulla. Again, you're just knowing the individual parts. You're not worried about diencephalon and brainstem. Oh, I forgot. That little thing right there is the pineal gland. I don't know if it's labeled up here. It's not, but that's your pineal gland. Oh, do we not have it labeled anywhere in here for you? Sorry, I'm just noticing this. I assumed it was in there. No, it is not labeled, so add that in. So we need to talk about the ventricles, and then when we're talking about that, I'm going to talk just a little bit more about the thalamus and the hypothalamus. For that, we need another picture, which I just made while I paused the recording because I realized I didn't have the images that I wanted to talk about this. This I just stole from our lecture notes. It's in there somewhere. It's a cross-section of a brain. If you look right here, those two little holes, those are your two lateral ventricles. If you were to cut further backwards in the brain, this is somewhere right about in the middle because there's temporal lobe and the back of your frontal lobe. as you go back, this is going to swing out that way. And if you moved forward, you would see it more out here. So three-dimensionally, it's kind of curved, and it curves out into the hemispheres. We'll go over where that comes from. And then this little thing right there, number 9, that is your third ventricle. And it's got this gray matter here on either side of it. Where it says 10, this circle of gray, that's the thalamus. And this 8 and 7 is hypothalamus. I don't know what the difference between 8 and 7 is, but 10 is definitely where the thalamus is, and down in here is hypothalamus. So what you're looking at over here when you're looking at the thalamus is actually just the inner wall of the thalamus. And if you cut right down the center of the brain, as this image is supposed to be, you're actually looking at the inside of the thalamus through a little ventricle that would be filled with cerebrospinal fluid. So that's what I wanted to mention about the thalamus. So to make sense of these ventricles, we need to remember the way the brain develops. So just bear with me, it's just ever so small an amount of neuroanatomy. Before you look anything like the biped, four-limbed animal that you are, you're really just sort of a flat sheet of tissue that's got like 3 layers to it. All they're showing you here is the top layer, but there's tissues underneath it as well. But the top layer is called ectoderm. It is what makes up your skin, and it's what makes up your central nervous system. So as you are developing from this flat-looking tadpole sort of thing, what was the neural plate starts to infold and becomes the neural groove, and you can see the groove is getting deeper here, and now the groove is deeper still, and it's starting to close, and eventually it'll close all the way up like it has here and form a neural tube. This is going to zip all the way down, and that's going to zip all the way up. If it doesn't zip all the way up, you get anencephaly, which is where you're missing higher brain centers. And if it doesn't zip all the way down, that I believe is spina bifida. So your whole central nervous system starts off as a tube, this neural tube here, which runs the length of what is going to become you. And then that tube starts to grow and get longer and also develop these weird folds. So diencephalon, this is where our third ventricle is going to be, because that's thalamus, hypothalamus. And telencephalon, this is going to form our 2 cerebral hemispheres. So this little bulge here and that bulge there is where our 2 lateral ventricles are going to come from. So what happens is your brain is, well, your whole central nervous system is the neural tube, but your brain is this whatever golden part, the purple, and then brainstem in green. And as it grows, it starts to get bigger, but your skull is forming around it, so it can't just keep growing in this direction. So what it does is it actually just grows in a circle like that, so that when you're done, you have ventricles that look like this. So you want to imagine that your brain was growing up, it hit your skull, and then it grew around in a circle. That's where your 2 lateral ventricles come from. And then your third ventricle is just what's left in the middle, cerebral aqueduct and fourth ventricle. I don't know if there are good pictures of this in our book. I don't think so, or they'd be in the lab guides. But I will show you where there's a model of this that is pretty much the same as the model that we have in the lab. So just looking at this right now, because it's up, right, this third ventricle, that is this space right here, and then those are your lateral ventricles. So that, if we look at it from the front, there's your third ventricle, and right here would be thalamus, and there would be thalamus, your lateral ventricles spiraling sort of through your cerebral hemispheres, and then cerebral aqueduct connects third ventricle to fourth ventricle, which is located, if we look here, just in between the cerebellum and the brainstem. And it is actually the cerebellum and the brainstem that form the boundaries of your fourth ventricle. When you look at it in the dissected brain, it doesn't look like anything because it just collapses when it's not filled with cerebrospinal fluid. If you want to know how to study that, it is anatomical models, nervous system, central nervous system. Oh, by the way, this also shows you, right, you can do the lobes. Practice anatomy lab will not do the gyri, the fissures, or the, I can't speak, sulci for you. But it will do the parts of the brain. Where is that? Here, it does different parts of the brain. There we go. Midbrain, pons, medulla. And then on the next one, I think it does this stuff, like thalamus, hypothalamus, pineal gland, all of that. So don't forget that that's in practice anatomy lab. But here are your ventricles. So lateral ventricle, boop, third ventricle, fourth ventricle, cerebral aqueduct you can't see unless you go to the next figure, and then its cerebral aqueduct is there. Lateral ventricle, third ventricle, cerebral aqueduct, fourth ventricle. And we have basically the exact same model which you might see on the lab exam. So those are the ventricles. And let's see, I think we want this one. Back to our list, just so I know what I'm talking about. Yeah, so that's all the parts of the human brain that you want to be able to identify on a model, most likely pictures of the model for the lab quiz, because that's what I've been doing. When it comes to the cranial nerves, it is simply this table that you need to commit to memory. You are not learning to identify any cranial nerves. However, the trick is that we number the cranial nerves using Roman numerals, which is the single dumbest thing I will tell you that you have to know. I have no idea why we still use Roman numerals for this. It's the dumbest thing in the world. I don't even know why we number them. We should just go by their names, because their names imply either what they do or where they're located. But this is life. I don't control the world. I just teach it. So let me show you one trick to doing this. If I can find, there we go, this is what I want. Let me just give myself a blank thing here, and we'll draw again. So there's just a, thank you, go away, there we go. What is it? A mnemonic, holy cow, I'm messing stuff up. Get with it, Dr. I. What's your problem? Here we are. Occasionally, our old trusty truck acts funny. Very good, sorry for my handwriting, vehicle. I just think that's a funny sounding word, vehicle. Anyhow. Right, so the first letter of each word is the first letter in each cranial nerve. So olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, accessory, and hypoglossal. Say that 5 times fast. I can't say it without help because I'm no good at memorizing lists. But this is a list that you just have to memorize. So commit this table to memory, learn your Roman numerals. Sorry, you still have to learn obsolete Roman numerals, right? It's this and the Super Bowl is the only thing we use Roman numerals for anyways. And I was just going to insult football fans and say, how many football fans know their Roman numerals? But I like football, so I can't insult football fans. Ventricles. So that's the stuff that you need to study. You need to do the dissection. So we'll talk about then what you need to do. So if you look at your lab report, I'm going to have to just open it. You are supposed to model a neuron out of clay. This is my example of it. It took me maybe 15 minutes to make. So you should be able to do this. If you threw away your clay, which somebody did in my other class, shame on you for wasting things. And I didn't tell you you could throw it away. You can just draw it. But if you make a drawing, it has to be really pretty and you have to use colors. So there. But this is my example of it. It's pretty straightforward. It does not have to be beautiful. It would be nice if you had different colors for your Schwann cells versus your axon, but the rest of it doesn't matter. Let's see, lab report. Where did it go? I thought I opened it. Apparently I didn't. Let's open that lab report. Here it is. Whoop. Oh, I see. It won't let me scroll up to the top. That is so strange. Fine, we'll just open it. There we go, lab report. Why, my mouse doesn't want to work today. There we go. All right, so sculpt out of clay, label these things. Bada boom, bada bing, easy. Then you're going to do your I can't speak today. Dissection, and I'm going to get rid of a couple of things for you because I eliminated them in the lab guide. So you can cross off mammillary bodies, cerebral peduncle. Instead of cerebral peduncle, you're going to learn midbrain. The external part of the midbrain that you can identify on the outside of the sheep brain is the cerebral peduncle, but we're never going to use that term in lecture. So just these things here are what you need to identify. Maybe if you do pins with tiny labels on them, or if you could annotate it digitally, whatever you can come up with. It doesn't have to be beautiful. And then I can go over how I would do it if I were you. So first, your brain shouldn't look like this. This is duramatter. It's very tough fibrous material. Your brain should look like this when you get it, no dura. So you should be able to see the lobes, and this little bit of duramatter right here should be gone. From the side, if it's intact like this, you just want to be able to identify the cerebrum and the cerebellum. And then on the underside, if you follow from the spinal cord, the first lump you hit is the medulla oblongata, the second lump is the pons, and these two lumps here are both part of your midbrain. Those are your cerebral peduncles. So on the lab dissection, I want you to call those midbrain. And if I want you to identify midbrain on the lab, it'll be when we do the sagittal section of the brain. So label these things if you want to on the outside. You really only need one picture of the brain, which would be this view with as many things labeled as you can label. Then what was I going to say? Oh yeah, how to do it. So right here where it says infundibulum pituitary stalk, on your brain there should be a tiny little hole there. It'll be a slit that runs from forward to back. That leads directly up into the third ventricle. right? And what you want to do, I won't go all the way up, like scroll all the way up in the lab guide, but you want to cut right through that third ventricle, so you're actually going in between the two halves of your thalamus rather than actually cutting through the thalamus. You want to cut through the third ventricle, which is in the middle of the thalamus. It doesn't have to be perfect, but all of the structures will look a little bit better if you're really close to the midline like that. So find that little hole on the bottom, go straight in, cut forward, straight in, cut back. The closer you can stay to the center line on the way back, the better it will be. And then when you get it in cross section, it should look something like this. And I think I can annotate this, right? Because I can draw with whatever that's called. I can draw in Word. So starting here, that's your spinal cord. Here's your first bump to there. So all of this is your medulla oblongata. Then you get your second bump from there to about here. This is your pons. And then right about here, I don't know exactly where to draw that one, and all of this, that's your midbrain. And then you can almost make out like a little dark gray area there. That's thalamus. This region down here is hypothalamus. So it's right in here where my neuroanatomy is a little rusty. I don't know exactly where thalamus and hypothalamus end and midbrain begins. But this lump right here is definitely midbrain. And that little groove right there that you can see, which there's more of here, that's your cerebral aqueduct, which runs right through the midbrain down to here, which is where your fourth ventricle would be, except this is all collapsed and you can't really see the fourth ventricle. That's a little bit of it right there too. Fourth ventricle. Let's see, so we did thalamus, hypothalamus. Where is it? I don't want to label the wrong thing, and sometimes it looks a little funky. So it's this little nugget right here is your pineal gland. This white matter here is already labeled corpus callosum. This piece right here, below your corpus callosum, that's your fornix. I believe that's on your list of things to know. Yeah, and that's it. So cerebrum, cerebellum, medulla oblongata, medulla, pons, midbrain, thalamus, hypothalamus, pineal gland, fornix, corpus callosum. This part here is actually some of the gray matter of the cerebral hemisphere. And that little groove right there, that's a little bit of lateral ventricle that you can see into. And here is a little bit more of lateral ventricle. There's the rest of the pineal gland. That's where it all went. That's nice and big. I was wondering why I was having such a hard time identifying it. There's your pineal gland. They didn't go exactly right down the center. And so sometimes it's easy to miss the pineal gland. I think that's it. I think that's everything I need to tell you and everything you need to know. So you're doing the dissection. Really, one picture of the cross section is all you need, or excuse me, the sagittal section. You're sculpting your neuron, and then again, after I'm done making this, there will be an additional link on the Brain Lab page to take you to mastering, to do some brain anatomy labeling homework in mastering. All of which will be due on the 21st. No, I take that back, the 22nd. Because I'm late getting this stuff up, so I'm going to give you till Sunday to get it done. There you go.